dear student, I am going to discuss the botany portion of the NEET 2026. Paper was simple. All these statements, if you see, they are by and large from your NCERT only, okay. So, flow with the paper the way I am explaining you, try to find out your mistake and try to calculate your marks, how much you are going to obtain in this paper, okay. So, let us move forward with the paper. 92. One thing from my side, if you look at question number 92, that doesn't mean it is that doesn't mean that I am skipping some question. That means 91 question was basically part of your geology. So, that is going to be discussed by the geology teacher. So, don't look at this number actually, just look at the question and the way we are discussing. Exploring molecular, genetic and species level diversity for product of economic importance is called. It's a direct statement from NCERT and the answer is your bioprospecting. Biomagnification is a word normally we deal with pollution. One of the classical example is cadmium pollution in case of some fishes we see, Minamata disease. Bioremediation again related with your pollution, and biofortification. You must have heard about this word in which we basically add some supplement or genetically modify some crops so that their nutrient supplements are going to be increased. It's a very wider term, biofortification. So, for this the answer goes with your bioprospecting. So, 93. Now, look at the question. Which of the following statements are true with reference to sex determination? In honeybee, if you remember, we have read in honeybee, it is haploid diploid type of sex determination. An offspring formed from the union of a sperm and an egg develops as a female queen. Fine enough. An unfertilized egg develops as a male by parthenogenesis. That is true. A male has half the number of chromosomes that of the female. That is also true. Males produce sperm by meiosis. Now, look at this statement. Got it? So, D goes to be wrong. And honeybee have a haploid diploid sex determination. Just now I told you when I started with the question. So, this is E. So, answer goes with A, B, C and E. That is your with respect to honeybee. Match list 1 with list 2. Growth regulator, function and effect. See, I will tell you one thing. This question is very simple. Can you see one thing? Herbicide. 2,4-D is a very famous herbicide. A3. Look at where A3 is given. Only one option is there. Answer is found. You are not even supposed to think also. 2,4-D, if you remember, there was one more 2,4-T also. So, there is only one option available here. And rest you can compare B1, that is GA3 is brewing industry. Then C4 is going to be nutrient mobilization, and ABA, that is abscisic acid, that is D2 goes with your stimulation of stomata closer. In racemose inflorescence, the growth is limited, the flower is solitary, the flower are born in acropetal succession, the main axis terminates in a flower. Now, see, in this condition, it's a straightforward question. This is not true, this is not true, this is not true. Answer goes with flowers are born in an acropetal succession. I hope you remember two words which you must have read, acropetal and basipetal succession. Try to find out the meaning of it. Another one, since the origin and diversification of the life on earth, there have been 5 episodes of mass extinction of a species. How is the 6th extinction, which is the in progress, different from the previous episode? Direct question from NCERT, direct statement. Now, let us read the statement. The current species extinction rates are far lower than those in the previous episode. This is a perfectly wrong statement. The present species extinction rates are 100 to 1000 times faster than the pre-human times. That seems to be true. The present net species extinction rate is 0. That is not at all true. If you see in the last 1 century, so many plants as well as animals have been become extinct. They have been lost due to so many factors. And the current species extinction rate is nearly 10 times faster than the previous episode. Now, if you look at this, You may be confused, with third and fourth, but this is a direct statement from your NCERT. The present species extinction rates are 100 to 1000 times faster than the pre-human times. Next one. The enzyme required for carboxylation in Calvin cycle is very simple, very simple. Hexokinase is in glycolysis, glucose to glucose 6-phosphate. Carboxypeptidase, not at all. What they are asking you is carboxylation. What does carboxylation means? You are fixing the carbon dioxide. RUBP seems to be correct. PEP carboxylase is not there because PEP stands for phosphoenol pyruvate. Try to recall where you have read phosphoenol pyruvate. So answer goes with your RUBP carboxylase oxygenase. Next one. Arrange the following in the correct developmental sequence related to microsporogenesis. Microsporogenesis means they are talking or they are asking you a question related with your pollen, grains and its development. Now look at the options. We will not look into the options, first we will try to solve actually. What are the words given? Microspore tetrad, sporogenous tissue, pollen grain, pollen mother cells. First is a sporogenous tissue. It will divide. It will give rise to PMC, that is termed as pollen mother cell. This pollen mother cell will undergo the reduction division, which will form the third one, that is termed as microspore tetrad. Tetrad are going to be holded by callose, which is going to be cut down into the fourth one, that is termed as your pollen grains. So can we find out the sequence? BD, 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 both are common. You can see here, this one is ruled out, this one is ruled out. Then comes B is here, D is here, then the third one is A and C. Answer is your fourth one. Got it? And pollen mother cell is diploid, pollen grain is haploid, this also you know. So this is going to be the sequence. Let's move on to the next one. In lac-operon, the Z gene codes for, if you remember the lac-operon was having RPO, regulator, promoter, operator and structural gene, Z, Y and A. I hope you remember. One is coded by this, one is coded by this, one is coded by this. So what you are trying to say is Z gene. So this will code for beta galactosidase, then you have the permease, then you have the transacetylase. So answer goes with your beta galactosidase. That is lac-operon. No. Match list 1 with list 2. Match list 1 with list 2. If you look into the options, I am not getting into the entire circle. This is something which is, can you see 1, 2, 3, 4, A. Now look at the another one I will give you. B also. 1, 2, 3, 4, all are different. So if you are able to catch just one of them, you will get the answer. No need to solve the entire thing. See this one. S phase, synthesis of DNA occurs and DNA duplicates. B third, where is B third? B third is this one, fourth one. No need to find out even, if you look at the answer in the first go, just find out which one is B or A, whichever you are comfortable with, you will get an answer. Not only this, you can see D also, you can see C also, everything is separate. Just one correct answer will give you the entire match the following correct. No need to match everything bit by bit. And we know S phase is known for doubling of the DNA, that is, the copies are going to be doubled. 2N2C to 2N4C, you must have read. Next one. The respiratory quotient of biomolecule used for respiration as per the above equation would be. So RQ you are trying to find out. What is RQ actually? RQ is going to be your none other than the CO2 evolved upon O2 consumed. If you look at the data, 102 upon 145. So if you look at this, It is not less, it is something in between 0.5 to 0.95. Got it? Because definitely it is not 0.5, because for 0.5 here it should have been something like 72.5. This is again not possible, because numerator and denominator, if you check, it doesn't go with the second, and 1 to definitely is not possible because both the datas are not same. So something between 0.5 to 0.95. Now they have not asked you the exact number, so that you are not supposed to calculate that. At least that entity had taken care of. You are not wasting your time in calculation. Just look at the option, look at the number which they have given you. Next one, let's see the next one. Which one of the following is not a characteristics of plant cell in the phase of elongation? New cell wall deposition, cell enlargement, large conspicuous nucleus, increased vacuolation. See what they are asking you, elongation. What does elongation mean? Suppose if you take the cell will elongate, it will grow up. Now for elongation, what you require? Cell wall must elongate, material must be added. Not a character, look at the question. Cell enlargement, that is but obvious, that is going to be there. Large conspicuous nucleus, let's put a dot on it. Increased vacuolation, definitely the vacuole is going to increase, So you are left with which option? Is not a characteristics of plant cell. Large conspicuous nucleus. Next one, let's see. Fine. List 1 with list 2, conjunctive tissue, Casparian strip, subsidiary cells, starch sheath. Now look at the very most simplest of all the cell, I will tell you, subsidiary cell C1 in the vicinity of the, so you are basically zeroing upon which one? These two options. D2 is already done, so D2 is common, so D2 is also common. This is done. See what I am trying to tell you is how to decrease the time even if the paper is lengthy. You are left with A3 and A4 and B4 and B3. Conjunctive tissue. Conjunctive tissue, if you remember, these are the tissue between the xylem and phloem. So C becomes your third, A becomes your third, answer is this one. If you remember, there is going to be a transverse section, there is xylem and there is going to be phloem, there is a cell here which is termed as your conjunctive tissue. Got it? So this comes out to be, see, what I am trying to do is, I am trying to tell you how you are going to eliminate the option. No need to match everything, it will save your time little bit. 1.7. In angiosperm, the root hairs arise from which of the following region of root? See, root cap zone, you know that there is nothing in the root cap zone. Root cap is basically a protective structure having graviperception material, and they help in penetration of root within the soil with certain enzymatic activity. Zone of elongation, meristematic, it will keep on dividing. Elongation, it is the zone of maturation from where the root gives rise to the root hairs. Move on to the next one. See, this is something very amazing question. This question is basically, if you open the NCERT morphology chapter, you open it, Solanaceae is there on your left hand side. On the right hand side, at the end of your morphology chapter, direct floral formula is written of Solanaceae family. So this is a direct formula. Okay, direct. You have, see, the difference is only this one. If you see, only this one. I hope you remember gamopetalous, gamosepalous, polysepalous, polypetalous. The concept comes from there actually. Which of the following is a triploid cell? What does triploid means? Triploid, basically when you say, that means you are referring to 3N condition. Look at it. Synergid, primary endosperm nucleus or cell, 3N, central cell, N plus N, zygote, 2N. I hope you got the answer. Because primary endosperm cell will only give rise to endosperm, and you know that the endosperm is triploid in nature. The only structure which is going to be the embryo sac after the fertilization, double fertilization, triple fusion, which will give rise to your triploid state, that is endosperm. Good. Decomposition, detritus, mineralization, humidification, everything is from a single paragraph of NCERT. Everything is from a single paragraph of NCERT and everything is written in a sequence. Now see, if you ask me, very simple. Humidification is accumulation of dark, amorphous, colored colloidal structure D1. Where is D1 in this condition? Fourth one. Even I am not reading the rest of the things. Even if I am not reading the rest of the things, you can directly get the answer. Can you see? First, simplest of all, humidification is simplest of all. You can see at this also, A3 also, that is going to be decomposition, breaking down of complex organic matter into inorganic substances. Then B is going to be 4, just 1 minute. Then detritus is going to be your dead remains of plants and animals, including the fecal matter. Let's move on to the next one. Fine. The main criteria used for 5 kingdom classification proposed by R.H. Whittaker 1969 included cell structure, cell organization, reproduction, and phylogenetic relationship. Biological classification, I hope when you open the NCERT, right hand side of the book, this entire paragraph deals, and it's a continuous line from R.H. Whittaker. Flagella is not the criteria, so where C is absent, C gone, only you are left with E, B, D, E. Next one. The evil quartet of biodiversity loss, including which of the following? Now, can you see in all the 4 options, there is something, soil pollution, water pollution, there is nothing in D, and air pollution. You know this fact very well that evil quartet doesn't include air pollution, soil pollution, water pollution, sound pollution, radioactive pollution. It's not included. Habitat loss, fragmentation, overexploitation, alien species invasion, and the last one is co-extinction. Third one. Move on to the next one. Arrange the following steps of DNA fingerprinting in a correct sequence. Isolation of DNA and its digestion by restriction endonuclease, hybridization using a labeled VNTR probe, transferring of separated DNA fragments to synthetic membrane, detection of hybridized DNA fragments by autoradiography, separation of DNA fragments by electrophoresis. Now, you all know that autoradiography is the last step. Now, isolation of DNA and its digestion by the restriction endonuclease. You take a cell, you cut the DNA, you isolate it. So, this goes with the first one. Then, once you have got the DNA, what you have to do? You have to separate it by the electrophoresis. Second one, A and E. Is there any A and E? Fine. 2 are still forcing us to read again. After separation, transferring of separated DNA fragment by synthetic membrane. Third one, is there any C? C. Now, B is which one? Hybridization using the labeled VNTR probe, and which is the last one, that is detection of hybridized DNA fragments by autoradiography. So, this becomes your A, E, C, B, D. They have not written all their steps, something they have missed out few of the steps, but anyway this is by and large written in your NCERT by flow. So, this is going to be the, and I hope you remember this technique, some blotting technique is there, transferring of separated DNA fragments to synthetic membrane. Try to find out which type of blotting is there, southern blot or eastern blot or western blot. Next one, which of the following statements are correct with reference to a transcription unit? If you remember in your NCERT, they have drawn a very beautiful diagram, is promoter, structural and terminator. 5 prime, 3 prime, 3 prime, 5 prime, coding, non-coding, then upstream, downstream. Everything is drawn in your NCERT. If you want, when I am discussing this paper, you can take out your NCERT diagram and just look at all the statement which I am going to read right now. A transcription unit in DNA is defined primarily by 3 regions: promoter, structural and terminator. 100% correct. Promoter, first one, then you have a structural, then you have a terminator. The promoter is said to be located towards 5 prime end of the structural gene, towards the structural gene 5 prime end. The promoter is a DNA sequence that provides binding site for RNA polymerase. Since you are talking with respect to transcription, so DNA will be coded in the form of RNA with the help of an enzyme that is termed as RNA polymerase, or you also say DNA dependent RNA polymerase. The promoter defines the template and the coding strand, that is true. You must have read about coding, non-coding part. There are 2, coding and non-coding, in your NCERT diagram, if you remember. The terminator is located towards 3 prime end of the coding, and it defines the end of the process of transcription, that is also true. So, is there any option with all the option? A, B, C, D and E. So, this goes with your 114. Which one of the following type of pollination brings genetically different types of pollen grains to the stigma? Clistogamous, a flower which never opens, that is clistogamous condition. Autogamy, self. Genetogamy, there is going to be 2 concepts involved in it. One is ecologically and genetically. Genetically is self, ecologically cross, I hope you remember. And then you have xenogamy, that is the last one, that will be 100% genetically, or you can say cross fertilization, which brings about the genetically different types of pollen grain onto the stigma. Which one of the following is an in situ conservation method? Now, when you look at the in situ conservation method, in situ means all the organisms are going to be preserved, conserved at the place where they are present. Seed banks we can create, so it is not an in situ conservation, it is a technically ex situ conservation. Sacred groups, natural condition in which the things are going to be preserved. Botanical gardens can be created, can be made. Okay. Then you have wildlife safari parks. This you know that this is not the in situ conservation. We define an area, we can create a safari park. So, this is sacred groups, that is going to be in situ conservation. Heterophilous development in response to environment is an example of which of the following phenomenon? Redifferentiation, dedifferentiation, elasticity, plasticity. Again, it is plasticity. Differentiation, dedifferentiation and redifferentiation is basically a concept related with your cell losing the ability to divide, regaining the ability to divide, and again losing the ability to divide. In this condition, they are not asking anything like that, they are simply asking the heterophilous condition. It is going to be the concept of your plasticity. Now, see this. Productivity, net primary productivity, gross primary productivity, secondary productivity, this is going to be there. Now, secondary productivity is something which is related with consumers. So, D2, C4, C4. Net primary productivity and productivity. Now, you know that the net primary productivity is nothing but gross primary productivity minus respiration. Net primary productivity is nothing but GPP minus the respiration. So, B goes with 1. So, answer is third one. Option can also give you the answer. Comes with the third one. In which of the following the ovules are not enclosed by an ovary wall and remains exposed? It is pinus. It is a 100% factual piece of information. And pinus is basically a gymnosperm, so not enclosed by an ovary wall and remain exposed. Which of the following statements are correct with reference to packaging of DNA helix? Histones are organized to form a unit of 8 molecules called histone octamer. Fine. Histones are negatively charged basic protein, not at all. Not at all. Histones are rich in basic amino acids because of arginine and lysine, true. So, A and C, where is A and C? Let's confirm D and E. The positively charged DNA, you know that the DNA is not positively charged. DNA is negatively charged due to the presence of the phosphoric acid H3PO4, which basically forms the backbone part. So this is ruled out. So you are going to have packaging, positively its D is gone. So A, C and E, correct. Next one. Match list 1 with list 2. I'll tell you one thing, it's a very simple. A2, there is only one option, just one option. Marginal is P, B is going to be exile, is going to be lemon, C is going to be parietal, that is going to be mustard, and basal is going to be your marigold. From only one you can get the answer, okay. Which of the following is the site for active ribosomal RNA synthesis? Not at all, not at all, not at all, nucleolus. Just think now how centrosome can synthesize RNA, or how kinetochore can synthesize RNA, or how chromatin material can synthesize RNA. Because for synthesis of RNA, you require an entire process, DNA to RNA. That process is termed as transcription. Lot of enzyme and everything is required. So the main function of bulliform cells in grasses: to perform the photosynthesis, not at all. To minimize the water loss during the water stress, that is the answer actually, but let's read the another one. To make the leaf impermeable for fungal spore, that is not possible, because fungal spore has a unique property of producing the lytic enzyme so that they can penetrate into the plant part on which they are growing, and they can penetrate into the plant part. So it doesn't work like that. To transport the water, that is a function of xylem. So answer goes with your second one, that is to minimize water loss during the water stress. Which of the following statements are correct? The Amazon rainforest being cut and cleared for cultivation of soybean is an example of habitat loss. Perfectly correct. Actually, on this, they had an United Nations also raising their concern that Amazon rainforest has been continuously cut, and because Amazon rainforest is considered to be lung of the earth, so the carbon dioxide absorption, or you can say conversion of CO2 into O2, the rate was going to decrease. It is decreasing actually. So United Nations has also raised this concern. Steelers cow, sea cow and passenger pigeon became extinct due to over exploitation by the humans. So A, this one seems correct, this one. So the Steeler sea cow and passenger pigeon became extinct due to over exploitation by the humans, correct. The Nile perch introduced into the Lake Victoria in East Africa helped in population growth of chichil fishes in the lake. It doesn't growth, downward trajectory it is. Water hyacinth is an invasive species, that is true. One of the classical example is in India, in which majority of the water bodies you will find water hyacinth. So A is correct, B is correct, D is correct, okay. When a species become extinct, the plant and animal species associated with it are not affected, not at all. If a species become extinct and it is associated with another species, there is going to be the extinction of the other species also, that is termed as co-extinction. So you have got A, B and D. Where is the option you have? A, B and D. I am speaking at length so that you can understand where you went wrong actually. I am not simply telling you A, B, C, D answer, okay. Which one of the following statement is not true about the universal rule of binomial nomenclature? When you look at the binomial nomenclature, you normally say it is going to be genus and species. Fine, let's find out. The first word is the biological name, represent the specific epithet, while the second component denotes the genus. Exactly opposite they have written. So this seems to be incorrect. See what they are asking. Not true. Let's go with this. Another one. The specific epithet is the biological name, starts with a small letter. A specific epithet with a small letter. Both the words in a biological name, when handwritten, are separately and underlined. That is true. This one is true. This one is true. Biological names are generally written in Latin, correct? So your option 1, because they are asking you not correct. Let's go to the next one. Which one of the following disorder is caused by substitution of glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin? Entire long literature is there in your NCERT. Answer is your sickle cell anemia. Phenylketonuria, hemophilia, thalassemia, they are genetical disorders due to one or the other reasons. Find the incorrect statement. Find the incorrect statement about the photosynthesis from the following. See, they are asking you incorrect statement, okay, about the photosynthesis. Water splitting complex or oxygen evolving complex, OEC also you call it, associated with PS1? No. PS2. C4 plant use the C3 pathway for CO2 fixation as the main biosynthetic pathway, correct? This pathway is common whether you take C3 plant or C4 plant, because ultimately fixation of CO2 is the job of C3 cycle. In C4 plant, photorespiration does not occur, correct? That is an advantage actually. You must have read about it. C3 plant exhibit Kranz anatomy, not at all. It is C4 plant which exhibit the Kranz anatomy. ATP synthesis in chloroplast occurs through chemi hypothesis. That is true. So we are searching for incorrect statement. So incorrect statement goes with first one and the so it is A and D. Next one. Match list 1 with list 2. See, I can do with only one option. Just with one option I can do. Polygenic inheritance, skin color. D1, where is D1? Now you can match rest of them. Incomplete dominance, codominance, pleiotropy. Before I say, you can also search. Incomplete dominance, where is flower color? Red, white, pink. Codominance, ABO blood group. In ABO blood group, only AB blood group is codominance, okay? AB blood group is codominance. O blood group represents basically your recessive concept. When A blood group is there, IA, I naught, that is also A blood group. That is also a concept of dominant and recessiveness. So they have taken ABO blood group, fine enough, that's not a problem. Pleiotropy, phenylketonuria disease in the humans. So I hope it matches with the third one. How many ATP and NADPH molecules are required to make one molecule of glucose through the Calvin's pathway? Straightforward. Straightforward. There is no if and but, there is no other calculation in this. Direct question it is. 134. Now let's check this one. Glycolysis, electron transport system, accumulation of proton, Krebs cycle. Glycolysis takes place in cytoplasm. So A3, let's find out where is A3. O. We have done it within the first line itself. B1, mitochondrial membrane. C4 and Krebs cycle. I hope it's fine. Next one. What is the probability of having a children with O blood group where both mother and father are heterozygous for A and B blood group? Let's do it. So what they are saying is mother and father are their blood group A and blood group B. Can you see one point in this heterozygous condition? What does heterozygous condition means? They both are going to be represented by IA small I, or you can also write I naught, that's not a problem. This is going to be IB small I. Fine. They are asking what is the probability of having a children with O blood group. So now let's go for the calculation. It will give rise to two different gametes, IA small I IB small I. This can go with this can go with this can go with this can go with this. Now look at the first combination, it is IA, IB. Look at the second one, IA small I. Look at the third one, IB small I. What is the fourth one? Small I, small I. What is the blood group basically? This is going to be AB blood group. This is going to be A blood group. This is going to be B blood group, and this is going to be O blood group. So what will be the percentage of O blood group? 1 by 4, 1 by 4, 1 by 4, 1 by 4, that is going to be 1 by 4 is nothing but 25%. Simple question. Let's move to the next one. Non-membrane bound cell organelles found in both prokaryotic and eukaryotic cells are. See this point. Cell organelle present where? Both prokaryotic and eukaryotic cell. And what is the condition for a cell? Non-membrane bound. And the membraneless organelle they are talking about. Lysosome. Not saying anything ribosome. I hope you remember 70S type and 80S type. Prokaryotic, eukaryotic, and they both are going to be, ribosomes are non-membrane or membraneless organelle. Lysosome have membrane, mitochondria is double membranous structure, centrosome also doesn't have. Ecological pyramid represents the relationship between the organism at different trophic level, and they are generally inverted for. Inverted, when we say inverted pyramid, that means the examiner wants to know about this particular condition. Now can you see one thing, one option you can immediately remove. Pyramid of energy is never inverted, it is always in the upright position. There is no exception for this. So there are three pyramids: energy, pyramid of energy, pyramid of biomass, and pyramid of number. Energy is never inverted, keep this in mind. Now, pyramid of number in grassland, it is again this condition, this condition. I hope you remember plants, then primary consumer, then secondary, then top consumer. I hope you remember. Pyramid of biomass in grassland, and they are asking about which one? Inverted. This is also upright. Pyramid of biomass in sea, phytoplankton, zooplankton and everything, if you can see, that is going to be inverted pyramid, and they are asking you which one? Generally inverted for. So this goes with your pyramid of biomass in sea. Because phytoplankton, zooplankton, they are very small. Even if you weigh all of them together, their mass is less than as compared to a bigger fish which is going to be at the higher trophic level. Next one, choose the correct statement regarding population interaction between the two species. Correct statement, okay. In both parasitism and commensalism, only one species benefit and other is harmed. Parasitism and commensalism, parasitism is, one is positive, one is negative. Commensalism doesn't go with that. Both species are beneficial in mutualism, positive. Both species benefit in commensalism, not at all. One is positive, one is 0. So B and A and C is gone, A is gone, A is gone, you are left with only 2 now. In parasitism, only one species benefit, other species is harmed. You can see here. So B and D and E will also become. Commensalism, one species harmed and another one is unaffected. So this goes with B, D and E. B, D and E. Now this question will take time because you don't have any other option. You have to read and you have to put your thought process there. Which one is amensalism, which one is commensalism, you have to put your thought process. Which animal do haploid cells divide mitotically to produce gametes? This basically is your male honeybee, haploid diploid system. Okay. Which of the following question depicts worst pearl logistic population growth? Direct question, dn by dt is equals to r into n, kn minus n upon k. That is a direct answer. Which of the following is an appropriate example of sexual deceit? It is Ophrys actually with bumblebee. This one is a type of interaction actually. Female wasp and fig, they lay their eggs. Cuckoo and the crow, again that is a type of interaction in which cuckoo lay their eggs into the crow. That is termed as brood parasitism, you might have read. Okay. Select the correct statement regarding the cell membrane in eukaryotic cell. Correct statement regarding the cell membrane in eukaryotic cell. Membrane of human RBC is approximately 52%. Look at the word eukaryotic cell. Now, can you see one statement? C. Extension of the plasma membrane into the cell forms the mesosomes. I hope you remember one statement that mesosomes are basically the characteristics of bacterial cell, that is prokaryotic cell. So, it is not applicable for your eukaryotic cell. So, wherever C is there, that is ruled out. Wherever C is there, that is ruled out. But you have to read that. Okay. So, answer is which one? A, B, D. Out of these four options, A, B, D is the most appropriate. Glycosylation is do present on the surface, but you can't choose because they are talking, because there is no such option available here. Glycocalyx or glycosylation is present. Anyway, with this option, it goes with A, B and D. Choose the correct statement regarding cell organelle and their inclusions. Cell organelles and their inclusions. Look at the first statement, what they are trying to say. The endomembrane system includes Golgi body, endoplasmic reticulum, mitochondria. Mitochondria is never the part of your endomembrane system. Ruled out. A. Rough endoplasmic reticulum bears ribosomes on its surface. That is true. Both mitochondria and plastid have circular DNA. True. That basically imparts them an unique character, that is being semi-autonomous organelle. A network of microtubule, microfilament and intermediate filament present in the cytoplasm is called cytoskeleton. True. So, wherever A is there, that is ruled out, gone, and BCD is present, BCD is present. Mitochondria is a single membrane bounded structure, not at all, it is a double membrane bounded structure. So, answer has to be B, C and D. I hope you got it. Okay. The sixth mutation of beta globin gene causing polymerization of hemoglobin and change in RBC is basically GUG. It's 100% factual information, 100% factual. You remember when you study this in case of your change in the RBC actually, that is due to the polymerization of hemoglobin. Next one. A population of a grasshopper species, the chromosome number of some member is 23 and some other members possess 24 chromosomes. 23 and 24 chromosomes bearing membrane in this species. Now look at this part. Grasshopper shows XX and XO type. XX and XO type. Not like human beings, which or Drosophila also you can include XX and XY type. In our case, XX is female, XY is male. But in this condition, what happens? XX and XO, that is going to be male and female. It is a good question actually. So, male and female. So, this is all about your botany paper discussion. Just go through the entire content which I have spoken right now. Try to find out your mistake. Try to find out what can be done in future. Try to calculate your marks. And if you see this entire question, all the question, if you go through our test series that we conducted in southern part of India, almost we conducted 30 test papers in almost 30 days. Every question that I have discussed over here was in one or the other form in that particular 30 series of the test papers. Not even a single question is out of the test series, not even a single question. They may not be a direct question, but one or the other format, the paper questions are there. So all the best for your result. Hope to see you again. Thank you.